Right, in this video we're going to talk about how changes in the genetic material affect living things. We're talking about mutations. So there are different kinds of mutations, but all mutations are heritable changes in the genetic information. They fall into one of two classes. Gene mutations, which affect individual genes, and chromosomal mutations, which affect the structure and or the number of chromosomes. So gene mutations include point mutations, which is a change in one nucleotide. And there are three kinds of point mutations: substitutions, insertions, and deletions. So a substitution is where one base is changed to a different base. So in the example here, a C is changed to a T. In some cases, a point mutation has zero impact because of the repetition of the genetic code. For instance, if you change UUA to UUG, the same amino acid leucine would be produced into that protein. So there's no effect. In other cases, it affects an amino acid, that one amino acid in a protein, which can affect the structure and function of the protein. In other cases, it may change a coding protein to a stop codon, which would have a much bigger impact on the protein. So you can see the example there. Substitutions, like I said, normally affect one amino acid. A silent mutation is when a substitution does not change the amino acid sequence of a protein. Usually it's the third base in the codon that's changed, and then you have this amino acid that's still the same, so no effect. A missense mutation changes the amino acid specified in one particular codon of a gene. A nonsense mutation would be change it to a stop codon, which would cause the protein to be stopped being produced, and which would most likely produce a completely non-functional protein. Insertions and deletions are the other two types. Insertions, when you add a single extra base to the code. Deletions, when you remove a base from the code. In this case, you change everything from that point on downstream because you've shifted the reading frame. Remember, the codons are read in sequences of 3. So when you delete one, it doesn't just affect that codon, but it affects every codon on down the line. These can have a much bigger impact on the organism. So because they changed the reading frame, we call them frame shift mutations. And then there are the chromosomal mutations, where the structure of the chromosome is changed. You have deletions where a section of the chromosome is lost, not just a single base, but a whole part of a chromosome. You have duplications where part of the chromosome, a section of the chromosome is duplicated. You have extra bits of DNA in there. Inversions is where part of the chromosome breaks off and reattaches in the wrong order. Remember, genes are read in a directional way, so that's going to have an impact on those genes in that inverted section. And then you have translocations, where genes from one chromosome are transferred to a non-homologous chromosome. So they probably don't have the right promoters and so forth to work properly. So mutations can harm, they can help, or they can have no effect. Harmful mutations obviously cause non-functioning or proteins that don't function as well as they should. Helpful mutations give new traits or change traits so that the organism has a better chance of surviving in their environment. That's the whole basis of evolution. And then you have the ones that have no effect. So some mutations are caused simply by a mistaken copy in the DNA. In other cases, mutations are the result of a mutagen, a chemical or physical agent from the body that changes the genetic information. An example, UV light will cause two thymines that are next to each other to get hooked together, so that when the cell tries to copy the DNA, it doesn't know what it's supposed to put in there. It just randomly puts a letter in there, a base in there, and oftentimes it's the wrong one. In other cases, you can have agents that can break and damage the DNA from high energy radiation, things like that. So some of the most harmful mutagens are those that dramatically change protein structure or gene activity. An example of this is the mutation in hemoglobin that leads to sickle cell disease. And it changes not only the blood's ability to carry oxygen, but causes these misshapen blood cells when the blood is under lower oxygen conditions, which can be the result of the mutation. And they tend to stick to each other and form blood clots and damage organs and things and cause all kinds of problems. Beneficial mutations are ones that can be useful, blend in better with your environment, maybe are faster, whatever. Polyploidy is a condition when an organism has an extra set of chromosomes. In animals, almost always lethal. But in plants, we have polyploid plants that are usually larger and stronger than normal diploid plants. In fact, most crops, many of the types of plants that we grow for decoration or whatever you want to call it are polyploid. And they become polyploid because we've actually done something to cause them to become polyploid, because of this, what we call hybrid vigor, where you have larger, stronger than normal polyploid plants. Like I said, most crops are polyploid.